Today I'm joined by Dr. Jeannette Tenthorey from the Hutch in Seattle. She is a new CRIM fellow for amfAR, and we're looking forward to learning more about her and her research. Thanks so much for joining.
Thanks for having me.
So first, why don't we start by learning a little bit in simple terms what your project is going to be for the next couple of years.
Yeah, so I think that many people at this point are pretty familiar with antibodies and these things that get stimulated by vaccines. But it turns out that your body's immune system has these much earlier defense systems in place to defend against viruses like HIV. And so these are systems that are there patrolling your body, ready for the moment when the virus tries to infect your cells. And it can be incredibly potent. It can be very effective at blocking infection from even happening in the first place. So these are really interesting to me from the perspective of trying to prevent HIV infection. But unfortunately for us, of course, HIV is very good at mutating and evolving, sort of similar to what we're seeing with SARS-CoV-2 right now, but even more so. And so it turns out that the virus is not actually sensitive. It's managed to escape from most of these very early lines of defense that we have. So really the goal of my project is essentially to understand why that is. Why is it that the virus is not sensitive to our early lines of immune defense? And then if we understand that, can we find ways to manipulate it so that they are or they do become sensitive to those defenses? So let me just be a little bit more concrete, maybe to help out with understanding that. So really my goal is going to be my research will be to use these new, really powerful tools that have been developed in the last few years to map and find all of the possible mutations in HIV that affect its sensitivity to some of these early defenses. And so by doing that, we'll be able to understand a lot about how these human immune defenses are supposed to work, what it is that they're looking for, and how they find the virus, how HIV has managed to escape them or not be sensitive to them, and then hopefully to find some drug therapies that we can use to kind of box the virus in a corner and make it so that it can't escape both drugs and our native human immune defenses at the same time.
So why don't you tell us a little bit more about the particular protein that's a part of HIV that these, I'm just going to say it, these restriction factors are seeking out?
Yeah, so I'm really interested in these restriction factors that are targeting the HIV capsid. So this is the protein that surrounds the viral genome and it protects it against a lot of different parts of your immune system. So it's really the stable shell that's kind of the first thing that your immune system is going to see when the virus enters into the cell. And so it's really a huge target for a lot of these immune defenses, which you so accurately called restriction factors.
All right. So it's a very interesting virus, but what made you get interested in HIV?
Yeah, I mean, for me, it's really just that HIV is so important. It's such a huge public health burden, even still. I mean, of course, in the US, but also around the world, there are many people every day who are still getting infected by HIV, many folks who don't have access to a lot of the therapies that have been developed. And of course, we're seeing some really alarming rises in resistance, HIV resistance to some of those therapies. So really, it remains a huge public health problem, and one where I think a lot of basic science research can help us to find better therapies and hopefully to make those therapies really long-lasting and to get a handle on really a long-lasting therapy that prevents HIV escape.
I know you're excited about training younger people to become scientists, but how did you decide to become a scientist?
Yeah, I will say two things. I mean, for me, even when I started learning about biology as a high school student or earlier, I thought it was so fascinating how these complex biological systems work, and they work so well, even though nobody made them, right? They weren't designed. They happened randomly and then evolved to get better, and they're quite complex, but remarkably effective. So I've always been fascinated in understanding how they manage to do that and how they evolve to do that. So that's really kind of the driving intellectual interest for me. But I will also say that both of my parents were scientists who worked in pharmaceutical companies. So they've been really trying to use their skills to find drugs and medical devices that can help with human diseases. And so they taught me to do what I love, but to do it to help people, to better the world. So that's been a big motivator for me.
Well, that's very cool. So, I mean, you've worked on different things in your career before this, but looking forward, how do you see your career unfolding in the future?
So you mean what are my future career goals?
Yeah.
Yeah. My plan is to start my own lab at an academic research institution. So that's for two reasons. One, so that I can continue to do this type of research to understand these restriction factors or early innate immune defenses against HIV. But also, again, because that will allow me to keep mentoring these younger scientists. So for me, I think that actually some of the biggest impact that I can make on HIV is not just applying my own skills and my own expertise, but really recruiting and training a lot of other very creative and talented people to keep working on this problem. So I think the more heads we can put together, the better. And so that's why for me, I think an academic lab is really, really important, is to meld that scientific research and training environment.
And how is the Crim Fellowship going to help you get all of that done?
Yeah, so you pointed out that I've done some other things in my previous career. So I'm actually pretty new to the HIV field. I did my PhD work in bacterial immunity and structural biochemistry within that setting. And so, I've been doing HIV for just a few years, HIV research, and unfortunately, a lot of that has been in the era of COVID, which has been very isolating. And so I really haven't connected very much with most of the HIV research community. And so aside from the fact that the Crim will give me the support I need to perform this research, but it will also give me a platform to really bring me visibility in the HIV community, to help me connect with those other researchers that have that advice and technical expertise that I think is really going to propel this research forward. So I'm excited about it from that perspective as well.
That's great. Well, with any luck, we're going to be going to real in-person conferences at some point soon.
Fingers crossed.
Right. And that will be a great opportunity for, us to get to know you better and you to get to know everybody else better.
Perfect.
Well, thank you very much for this chat. It was great to get to know more about you and your research. Wishing you all the best in the future. Thanks so much.
Thanks a lot. It was great to chat.